Hi everyone. This is a quick overview of lesson 4 and some of the main content in it. In this lesson, we're looking at how viruses make us sick. So we're going to go over the viral structure as well as the viral life cycle, and then talk about how viruses can change over time. Again, as usual, you'll have the main overview of the lesson in Canvas, and your notes are being filled in right here in your OneNote. So the very start of the lesson is looking at an actual size of a virus cell. So you can see the average eukaryotic cell here being about 20 micrometers, and then comparing that to the size of bacterial cells, and then comparing the size of a bacterial cell to the size of a virus. So you can see that viruses are very, very tiny, and viruses can actually infect bacterial cells as well too. And so remember that viruses need a host for them to be able to function. And so they can infect basically any living cell, plants, animals, bacteria, fungi, you name it. So we start the lesson off by looking at what are viruses, and simply it's a small infectious agent. It cannot reproduce on its own. It cannot make proteins on its own. It needs a host to be able to do that. And you'll see in this lesson how they do that, but essentially they inject their genetic material into the host, and the host does all the work for them. So viruses can either be DNA or RNA viruses, and depending on the type of genetic material, it will depend on the enzymes that a virus needs or can use in a host cell. But we'll take a look at that after. So, what does a virus actually need to replicate? Viruses are pretty simple in terms of their structures. They have 3 basic structures. Some only have 2 of them. The first piece is the genome. So the genome being your genetic material. And again, we mentioned that can be DNA or RNA. The second is the capsid. And so the capsid is simply the part that protects the genome. So in this naked virus, the capsid are these kind of little pink blobs on the outside here. Whereas in this envelope virus, this would be the capsid right here. These pink blobs protecting the genetic material. Now, hopefully it's pretty self-explanatory. The envelope is only going to be found in envelope viruses. Naked viruses do not have an envelope. So the envelope serves as an extra layer of protection. It's a lipid protein and it covers the capsid. Okay, and we'll go over some common examples, but again, you can probably tell here that envelope viruses are going to generally be more successful because of that extra layer of protection. So in this chart here, it goes over the main functions of these 3 structures and what that looks like. In these pictures here, this naked virus is something called a bacteriophage, or phage for short. And as it sounds, bacteriophage is related to bacteria because these are viruses that infect bacteria. And this right here is actually an example of the influenza virus. And you can see it has that envelope here. But in each case, it's showing you where the genome is, where the capsid is, and the envelope, if applicable. So you have that chart here. You're going to use that to fill in your notes about the overall function of those 3 structures. And then I want to get into actually drawing out your viral life cycle. The very first part of that, though, is draw a simple example of a naked and envelope virus. So you can see my example here. I just drew a really quick example of what that looks like. Make sure you label your drawings. And I've only partly drawn in here some of these cell surface molecules, these proteins, that are actually really crucial. We'll talk once we get into the life cycle about what those do and why they're important for envelope viruses. Okay, and once you've drawn that, there's a little Edpuzzle video for you to complete. So you can pause this video here. It's about 6 minutes, but it's good. It'll give you an overview of what the life cycle looks like. So pause this video, and then we'll go over the steps of the lytic cycle. Okay, so hopefully you finished the Edpuzzle video now, and we're just going to go over a quick overview of the steps of the lytic cycle. So the very first step is the attachment or entry step. And if you're a naked virus, you can do this one of 2 ways. The first way is pretty easy. The virus would latch onto the cell membrane of the host and inject its genetic material through the cell membrane, and then use the host's enzymes to replicate its genetic material and make those viral proteins. One of the examples of viruses that can do this are bacteriophages. So again, they would latch onto the cell membrane of the bacteria inject the genetic material in, and the bacteria would do all the replicating of that genetic material and then making the new viral proteins as well. The second way is that a naked virus can bind to these host cell receptors and be swallowed or engulfed into the cell through endocytosis. Once it gets into the cell, it actually punches holes in the endosome, that kind of vessel that took it into the cell, and then it's free to kind of roam around in your cell, and then it can release its genetic material. And in cases of eukaryotic cells, that genetic material ends up going through the nuclear envelope in the nucleus, and our enzymes will do the replicating for that virus. And an example of this, like shown in this picture, would be the poliovirus. So in your OneNote notes, I just want you to draw a simple example of that. You can draw an example of either the injection or the endocytosis step. It's totally up to you. So in my really simple example here, I've shown that injection step. Here's my bacteriophage, and it's injected its genetic material into the host cell. Okay, so again, you can draw either or. In an envelope virus, there's really only one way to go about that, and that is through endocytosis. So because an envelope virus has these cell surface proteins on the envelope, those proteins need to be recognized by the host cell. And so again, you can see these kind of host cell surface receptors, and think of this as like a door and a key. If the key fits, it'll be allowed entry into the cell. And so you can see it being taken up here by the endosome. Okay, and once it's taken in, you'll see the cell surface proteins will stay at the top of the cell membrane, and just the capsid and the genetic material gain entry through the cell membrane. And that's an important step. You'll see that again at the last step when the virus is exiting the cell. Again, once it's in, then the capsid will start to disintegrate, and it'll release that genetic material. And once that genetic material gets copied, it'll make more capsids, more proteins, and then can start to release. So draw that in your OneNote, those simple steps there. And then we go on to step 2. And step 2 is all about hijacking the host, getting the host to do the work for you. So like we mentioned, your viruses can either be DNA or RNA viruses. DNA viruses follow this idea of molecular dogma, that you start with DNA, it gets transcribed into RNA, and then translated and formed into proteins. In an RNA virus, it works a little differently because you're starting with RNA, and then you actually make DNA from that RNA. And then from that double-stranded DNA, you're making another strand, of RNA, and then that gets converted into your proteins. And to be ordered to do this, the RNA viruses need to bring in specialized enzymes because our host cells can't go from RNA to DNA. And so I wrote a little note down here, and it just says they have to bring specialized enzymes. If you're in grade 12 bio or you're taking it next semester, you'll learn more about some of these enzymes that we use for DNA replication. Anyways, that's the second step right there. I'm not having you draw that. I just put that kind of idea in, that DNA virus is a bit more simplistic because you can use the host enzymes to do all that work, whereas RNA viruses, they need to bring in some specialized enzymes to be able to go from RNA to DNA. Okay, and in the last step, this is the exit step. For naked viruses, it's pretty simplistic. It literally will fill the host cell into the cell is so full of these new viruses that it bursts, and that's called cell lysis. So you can see here the host cell, this bacterial cell is bursting with all these bacteriophages, and then they can go out and spread to infect a new cell. Envelope viruses looks very similar to that entry step, but kind of in reverse. So you can see those viral proteins that were stuck there at the cell membrane. Now you have these new assembled capsids and genetic material that'll come up to the cell membrane, and it will collect here. You can see it's starting to form again and simply bud off. So it's taking all those proteins left at the cell membrane and is reforming that envelope and budding off the host cell. So it doesn't necessarily kill the host cell because it's not causing it to burst. And then it can go off and infect new cells from there too. Okay, so in the last part of the lesson, we're looking at viral evolution, so how viruses are capable of changing over time. I want you to watch this video first before we go in. So pause this video, and then we'll go into more depth. Hopefully you actually did pause the video and watch that. If you did, you should have an idea of what antigenic drift and antigenic shift are. I'm just going to go over it quickly, and these pictures right here are really good examples of what that actually looks like in viral cells. So with antigenic drift, this is the reason why we have to get a new flu vaccine every year. If you remember back from grade 11 bio, you'll know that mutations in genetic material are random. And if a cell, a virus, mutates, it can result in the production of new proteins. And so in the case of these viruses here, it's showing that the proteins or the antigens on the cell, on the surface of this virus, I guess it's an envelope virus, are changing over time. And so the antibodies that your body had before that were used to recognize this virus and we could fight it off, when those mutate, they're no longer, your body, your immune system is no longer able to recognize it. So we talked about this earlier back, like in the beginning of the unit, and this is an example of your influenza virus. And so a lot of these influenza viruses are named H number, N number. So H1N1 is one of the common examples. But because you can have all these different shapes and sizes of these antigens or cell surface proteins, that changes the number. And so one of the antigens is in relation to H and one is in relation to N. The H is hemagglutinin and the N is neuraminidase, just two names of the proteins that can change over time. And so there's H1N1, H2N2, H3, N13, whatever. There's a whole bunch of different combinations. And so because viruses can change and they rapidly replicate inside a host, you can have a lot of these mutations occur over time. And the influenza virus, which can affect a whole bunch of different species, that also plays a role in it as well too. So that's antigenic drift, the mutation that happens inside the viral genetic material. Antigenic shift is basically when you get mixing of viral strains. So in this example here, you're looking at bird flu and human flu. And you can see that a pig ends up becoming infected with both human flu and bird flu. And so this kind of idea of antigenic shift is much more common now because humans work more closely with animals in terms of farming, markets, pets, things like that. And so this happens much more commonly where a host like this pig can become infected with more than one strain of the flu. And inside the cells, the genetic material can mix, and that can result in a new type of a new strain of the flu virus. And so this can be very dangerous as well too. And this is one of the reasons why you can get pandemics happening, because it's this real like jumbled mess of different proteins from different strains of flu viruses. And as it mentions here, it can be two or more different strains of a virus, or it can be two or more different viruses like in total. So you can have the flu virus mixing with the rhinovirus or something like that. And so it really causes complications and causes us to constantly have to adapt to these ever-changing viruses. So back in your OneNote, make sure you're answering these questions after you watch the video, and then you're just outlining the differences here between antigenic drift and shift and providing an example to communicate your knowledge. I hope that helps. If there's any questions, please make sure you post in the Teams chat. Thanks.